I ask ladies, please, with the conversation. So we have oxidative phosphorylation. What does that mean? Why do we call it? What is phosphorylation? What do you think's happening there? That's right. Adding a what? A phosphate. When do we add a phosphate in all this that we've been discussing? When you make ATP. So that's what that word means. So why do we call it oxidative? So it has to do with oxygen. So we're using oxygen. How do we use oxygen gas in this process, in this oxidative phosphorylation, which is what? The electron transport chain, right? Which makes water. But how are we using? How is oxygen being used though by the process? What's its function?
Does it bring it
All right. Wow. Stop. Oxygen is a waste product of photosynthesis. Please separate the two things in your mind. Photosynthesis, respiration. We're talking now about respiration. Oxygen is a waste product of respiration, but photosynthesis. But in respiration, it has a function. What is the function of oxygen in oxidative phosphorylation? Good. Last electron acceptor. Very good. The last electron acceptor. That's why we call it the electron transport chain. That's what we're talking about, the ETC. We call it and that's happening in the mitochondria. And this is why we call it oxidative phosphorylation, right? Now, this whole thing is separated into two parts, remember? We have the first part, we're coupling it. Remember someone said coupling? I'm recording it, so I'm not saying the person's name. What did I say about the jean jacket? Please? Can't you just take it off? Oh my gosh. So oxidative phosphorylation. So we have, what was I saying? So oxidative phosphorylation is broken up, or ETC is broken up into two steps. First is chemiosmosis, right? And in chemiosmosis, what are we doing?
Moving the hydrogen.
We're from low to high. We're making low. We're going from low to high.
I thought it was high to low. You said high to low. You said high to low.
Jesus, give me strength.
You said high to low. Yeah, he said low to high. No, it's not.
May all the powers of the universe help me go through this. Chemiosmosis is going from low to high. It's creating. It's the definition of chemiosmosis. It was in your homework. It was one of the things you had to do is define chemiosmosis. It's in your chapter 8. I've said it several times. Go back and reference the videos. If I said it incorrectly in a previous video, call me on it. But until then, let's move on. Creating a concentration gradient. In other words, we're creating what? A high side, high hydrogen ion concentration on one side, and a low on the other. And we're creating it by doing what? By pumping them from low to high. How do we do that? We use what? We use proteins, is correct, but we also use electrons by using, right, the proteins everyone should know. It should go without saying almost at this point. But we do it by using electrons that we got where? From where? Glycolysis and what? The Krebs cycle. And remember, that could be a citric acid cycle. I don't know what they're going to put in the course exam, but it could be citric acid or TCA. But it's a Krebs cycle is what we're going to call it. So that's chemiosmosis. The second part is we're going to create ATP, right? That's the whole purpose. That's the whole purpose of the ETCs to create it. But we had to do all this first so that we can make this, right? We had to create the concentration gradient. That's the first step. We had to create the concentration gradient. That needed to happen before we could make ATP. We used an enzyme called ATP synthetase. All right. We used the concentration gradient. Right? Made by what? Chemiosmosis, very good. Which depended on the electrons, which came from where? Where are the electrons? From TCA and from glycolysis, which originated where? Where was the where did these electrons come from? Where? Yes, that's right. But originally from glycolysis, and originally from what molecule? Sugar. So all these came from sugar. And it could be another biomolecule, but let's just stick with sugar for now. So create ATP synthetase. It uses the concentration gradient and of course it uses ADP, right? Plus phosphate. And by the way, oxygen, I forgot over here, oxygen gas is what? The last electron acceptor. Do we all agree with that so far? Yeah. The last electron acceptor. Uses ATP. And, oh, what the heck. And because, and let me just put this on the side, I'm going to put a little asterisk on here. Because oxygen gas is used, right? We call this oxidative phosphorylation. And this is kind of the factory line model for making ATP. We make a lot of ATP here. See, 34 ATPs made. 34 to about, I'm going to put about, the number can change a little bit, but about 34 made versus 2 in glycolysis, versus 2 net in glycolysis, 2 net in Krebs cycle, right? So there's not a lot of ATP made in glycolysis in Krebs cycle. You get all the ATPs, a lot of ATPs here in the electron transport chain. So this is what makes the mitochondria so good at making ATP. Are we good with this? This is respiration. This is a summary slide for respiration, right? You should know all this for the test, and you should know where this is all happening. We've talked about it. The rest of the video talks about that. Let's go on now and talk about fermentation. So you have glycolysis, and we talked about it's happening where? That's right, in the cytoplasm. So this is happening in the cytoplasm. And what's the end product of glycolysis? NADH. That's right. NADH is one, and pyruvate is the other, right? So those are, besides a couple ATPs, it's true. Question?
In the last test we had, they said, like, where was the glycolysis, and they said somewhere, they said, like, it was surrounded by a fluid in the cytoplasm. So I'm just wondering, can you be more specific of where it is at the cytoplasm?
All right. It's happening throughout the cytoplasm. There's no real one place. It doesn't have a spot where it happens, at least not that I know of, okay? It's happening throughout the cytoplasm, wherever the enzymes come together for it to happen. So this is a biochemical pathway. As far as, to my knowledge, there's no like region of the cell where this happens. I'd like to tell you that it happens right outside the mitochondria, but I'm not sure there's any specific region. We know that the TCA cycle or the Krebs cycle and the electron transport chain both happen inside the mitochondria. Question: in photosynthesizing eukaryotic cells, where do all the reactions for photosynthesis happen? Chloroplast, right? So if you have a regular, there's exceptions to everything, but remember that in cellular respiration, things are happening both in the cytoplasm and the mitochondria. So you have this glycolysis going on. We're producing, what is it we produce? 2 pyruvates. Now remember, glycolysis, that means what's coming in? Sugar. Now there are other places that things happen. I don't want to really get involved in it too much, but we know we're making a net, 2 ATPs, right? I notice I'm keeping it very, very general. And we're also making some NADH, right? And what is NADH doing? It's an electron transporter. So it's transporting electrons. Under normal circumstances, with plenty of oxygen, it's going to be going to the electron transport chain, right? But what happens if there's no oxygen? Under what conditions do we not have oxygen? Under what conditions do we not have oxygen to our tissues? Speak up, I can't hear you. Well, no, I just asking you, when do your cells not have oxygen? When you're exercising a lot, very good. Like Mr. Krupa is a marathon runner, right? So when he's running a marathon, he's under anaerobic conditions because as much as his heart's pumping and he's breathing really hard, his body still doesn't have enough oxygen, right? That's why you breathe faster, because your body's trying to get what? Oxygen. More oxygen, right? That's why when you guys go up the stairs, you're trying to get that oxygen because your muscles are being challenged. If your muscles aren't being challenged, you're not breathing heavily. Does that make sense? So that's one very normal way that happens to all of us all the time, at least those of us that exercise at all. I do some, not enough, obviously. All right, so pyruvate. Now, if there's no oxygen, there's nothing to take the electrons. So now you got a problem, right? You're going to run out of NADP because remember, in order to get NADH, what do you need? You have two of these, NAD plus. You need NAD plus to make NADH. Does that make sense? Because this NADH is NAD plus is taking the electrons with the hydrogen and making NADH, right? NAD plus is taking the hydrogen and the electron and making NADH. So if you don't have enough oxygen, the electrons back up and there's not enough NAD plus. If there's no NAD plus, glycolysis stops. That means no ATP. That means cell death. So this is enough ATP to keep you going for a short time, right? That 1 mile run, the reason you just don't stop and die is because you're producing enough ATP to keep your bodies going. Does that make sense? So I'm first talking about humans, then I'll show you what happens in yeast and in yeast or fungi and in some bacteria. All right, are we going to go? And now watch. What happens, you can take a pyruvate. You do not have to know the name of this molecule, but I'm going to tell you. Well, I guess you should know this molecule. What you're going to do is, by a process, you're going to take and you're going to deliver those electrons to what? To this process. What do we call this process? Fermentation. Fermentation is what happens when you don't have oxygen. This happens when you have anaerobic conditions, or you know what, I'm just going to say without oxygen. Gas. Is everybody okay with that?
Yeah.
No oxygen, gas, you do fermentation. Your muscles do it, yeast do it, bacteria do it. That's what happens when you don't have oxygen. You have to keep the system going. You have to have something to take that hot potato. You have to have something to take that electron. And in this case, it's fermentation. Now, as a byproduct, you produce this stuff called lactate. I just spelled it. Lactate gets turned into lactic acid. Lactic acid's no good. Too much of it, it'll poison your cells. But at least you get some ATP. So if you're running a few miles, you're going to start producing lactic acid in your muscles. It's part, not the whole reason, but part of the reason you get pain in your muscle, in your legs. Do you ever run and your legs feel, not Charlie horse, but you know how you get that soreness? Not afterwards. After the next day, it's because your muscles are torn and they're healing. But during the run, did you ever feel like your legs are like, ow, man? You ever run long? I used to, in college, I ran long distance. You feel it, right? That feeling is there's a buildup of lactic acid in your muscles. That's not a bad thing. That's how you keep your muscles alive. But you don't want too much of that lactic acid buildup. Luckily, after you rest, you get enough oxygen, you start to change this back to pyruvate. And then pyruvate can go into the mitochondria and you can make ATP. So luckily, this is not a real big problem unless you're trying. It probably was a problem to that first marathon runner. You know what happened to the first marathon runner?
No.
The story goes that it was in Greece, and there was a war, and he was being sent back to the king to tell him that the general sent a messenger to the king to let him know that the troops needed help. He ran, something like 20 miles back to the king. He delivered the message and died. Because he ran 20 miles, probably, probably just. So by the way, that also tells you if you're going to run a marathon, you should train. But all right, so lactic acid may have had something to do with that, but in any case, the point is that lactic acid buildup is no good, right? So when you relax, when you rest after your run, I'm not trying to tell you exercise is bad. I'm just saying the extreme is bad, right? So after you rest, the lactic acid gets converted back. You get the arrows go in the opposite direction, back to pyruvate. That's it. That's fermentation. Now, that just happens only when you don't have oxygen. Now, there's another kind of fermentation that's important because as you know, our society drinks a lot of alcohol, beer and wine and etc. All that. Huh? There's another type of fermentation that bacteria and yeast do, and when you bake bread, yeast also produce this. The difference is when you make the bread, it's hot, so the alcohol burns off, but they're also producing some alcohol. Anytime you use yeast, there's a possibility of making alcohol. Alcohol, as you know, is a poison. You know it's a poison. How? How do you know? That's right. It's used to kill germs because it's a poison. So alcohol is not very good. Like lactic acid, alcohol is not good for the yeast either. If you let yeast grow, what we do with the yeast, by the way, is we put a bunch of hops. You can put honey in it, and you can ferment honey. You can put rice in it, and you make sake if you use rice. If you use honey, you make mead. If you use hops, you make beer. If you use grapes, you make wine. But no matter what, all these yeast are going to take these sources of glucose, right? And they're going to, without oxygen, so they seal it in an oxygen, in a vacuum, not in a vacuum, but in a place where there's no extra oxygen being used, available, so that they ferment. So they put it in a wine cask. You ever see those big giant wooden barrels of wine? They just leave them there. Yeah, question.
So what you talk about, like yeast that they put in cakes and stuff?
Wait, what did you say?
He said yeast in like cake.
You put yeast in bread.
You put yeast in cake, anything, yeah, in cake too. And some people do, anyways. It depends on the type of cake, I think. But the problem I'm not a baker, so I wouldn't know. But I can tell you that anytime you want, you can get the bread to rise without yeast. But most of the time, you use yeast to make the bread rise. So bread really is a big source of rising. I'm not sure if they use it for cake or not. But they make little air bubbles. You know how you cut into bread and you see all those little pockets inside the bread? Those pockets are because of CO2, right? Because you're producing CO2, right? They produce the carbon dioxide actually pushes the bread out and makes a little bubble for itself. The yeast do. But you also make this, ethanol. Ethanol is the alcohol that is in wine and different kinds of alcoholic beverages. And so this is called alcoholic fermentation. So this is lactic acid fermentation, right? That's what we do. Yeast, etc., do alcohol fermentation. And that's it. That's all there is to it. Now we have a choice. It's 9:47. Do you want to do a pop quiz? Or do you want me to kind of go back and just outline the whole thing?
Outline.
Raise your hand if you want to outline the whole thing. Raise your hand now if you want to do a pop quiz.
Wait, don't the pop quiz are great?
I think if you're okay, we're wasting. Well, I don't want to waste time. I need you guys to make your decision. So you made your decision. We're going to go on then. What we're going to do then, let me quickly review what we've been talking about and kind of outline what we did, and you can go back and look at it. So in a plant cell, now we're in a plant cell, you have two major organelles that have to deal with energy. First is a chloroplast.
You started over from the beginning.
From the very beginning,
For photosynthesis.
I'm going to do both. I'm going to outline the whole thing. Outline means I'm not going into details. Please don't forget that. Over here you have what? A mitochondria. Now, of course, in the chloroplast, you have the thylakoids, you have the granum, you have all that. There's different spaces, right? So the membrane. So I'm just going to draw a membrane, if you don't mind. I'm going to go ahead and draw a membrane, and I'm going to use that membrane as our example. So you have to remember that there are thylakoids and there are granum and there's spaces, but I'm just going to use a membrane. So here's this long membrane. And along this membrane, the first step, of course, is the light reactions, because you're going to have a photon coming in, hence light reactions, right? And the first thing you have is this little antenna-looking thing. It's an antenna with a center, has little hydrophobic tails on it. What do we call this? This is the antenna, right? Pigment. And at the center is what? Is the reaction center. So the light's going to come in and bounce around here. The energy bounces around all in here and then goes to the reaction center. At the very same time, what are you getting? You're getting water, H2O, going in. Another enzyme breaks the water up into what? Oxygen gas. Of course, you need two of these, right? Two waters. Plus hydrogen ions. So two waters, and it does that twice, obviously. And so what's it taking from water? It's taking the electrons. So now we got the electrons in the system. The photon excites the electrons, and that's how you pass that electron around. But then you pass it on to different proteins that are embedded in the membrane, right? And what are these doing? What are these proteins doing? This is what? What are we talking about here? Photosystem 2, right? That's the first one, photosystem 2. So as the electron gets passed around, I'm going to keep it yellow, gets passed around, what's happening in photosystem 2? Electron's energy is coming down, that's correct. But what's the purpose of passing them around?
Oh, it's it's the hydrogen ions.
Exactly. This is the chemiosmosis. So chemiosmosis is happening over here too, as you were testing on that. That does not mean, remember, water is osmosis, right? So the movement of water is osmosis, diffusion of water is osmosis. But chemiosmosis is not the diffusion of water, it's the movement of hydrogen ions to create a what? A high and low. It's from low to high, but it's to create a high and low. A high and low. Maybe that's where you guys are getting it. All right, so that means the hydrogen ions are coming in here like this, right? Every time electrons pass, the hydrogen ions coming in here. So why would you do that? Why would you even do that? Why do we do chemiosmosis? To make ATP, because we're coupling it, remember we're coupling it with another and with an enzyme, what's the enzyme we're coupling it with? ATP synthase. So we're gonna couple it to ATP synthase. And what that allows is the hydrogen to go from here through, allowing the making of what? ATP. So ADP plus inorganic phosphate comes in and you make ATP. This is not oxidative phosphorylation, right? Because there's no oxygen used here, right? What's the last electron accept? Well, we can't talk about the last electron acceptor because then in most plant cells, what's going to happen is that the electron's going to get passed to what? Photosystem 1, right?
Yeah.
And in photosystem 1, what's going to happen is you're going to have another photon come in, right? It's going to excite that electron again, and it's going to pass it. I told you don't need to know the different proteins in the membrane. What I do want you to know is what? What's the last electron acceptor in photosynthesis?
Oxygen.
No.
NAD.
Oxygen is not the last time. Oxygen's gone. That's right. NADPH. NADP, right, comes in and makes NADPH.
Yeah.
That's the last electron acceptor in photosynthesis. NADPH. All right. So but notice that it's the same process, isn't it? You're still using an electron transport chain. You're still using a photon and a photon. You're still passing the electrons around. The key is you need that NADPH because you're going to use that where? Out in the stroma. In the stroma, you're going to use it where? This is all in the membrane. In the Calvin cycle. And the Calvin cycle, what are you doing? What are you going to use? You're going to use the NADPH. That's right. Good. What's the carbon source? CO2 is the carbon source, not sugar.
Yeah.
And it's going to go into the Calvin cycle. Along with what else? What did you make here? What is this? ATP. That's what your whole purpose is, to make ATP. Why do you need to make ATP? A lot of it is because this also needs ATP. So the three of those things come together to make what? To make what?
Sugar.
They go to the Calvin cycles, right, to make sugar. Now sugar, the plant can use sugar for a lot of different things, right? But one of the things that you can do with sugar is out in the cytoplasm you can do glycolysis, right? And in animals, this is the only thing you do. In plants, you can do both. Glycolysis is going to break the glucose up into two pyruvates.
Wait, I thought that was
Glycolysis. You right?
Glycolysis. It is called glycolysis. I'm just saying glycolysis so that you remember it's glycos means sugar, lysis means break. I'm just trying to emphasize the definition. But you're right. It's glycolysis. So you're making two ATPs net, right?
Yeah.
And you're making two NADHs. No, bless you. There's NADPH is only in the chloroplast. Outside is NADH, and in animals it's only NADH.
I thought the net was two.
Huh?
Oh, never mind. Cause never mind.
All right. So that's what's happening here in the cytoplasm. Then in the mitochondria, oh, what the heck? What's going on with this? Over here in the mitochondria, the pyruvates, there's more energy. If there's no oxygen, and you're talking about an animal, certainly in yeast and bacteria, you have fermentation. So there's a process for animals, right? And yeast and bacteria. But in everywhere, in both plants, animals, and bacteria, and some bacteria, you have the pyruvate goes into the mitochondria to where? The Krebs. I put Krebs.
I was gonna say, what's Krebs?
The Krebs cycle. And remember, it's a cycle, so all this stuff is really generated over and over again. But before pyruvate doesn't go directly into the Krebs cycle, first thing happens is what happens to the 3 carbons in pyruvate? They get turned into what? CO2 gas, and that leaves. Now, if it's a plant, the CO2 can come right back into the chloroplast. If it's an animal, the CO2 gets expelled out, right? But CO2 needs to go. The electrons, though, they go into the into the Krebs cycle via any, all the rest of the stuff that has to happen, and around it goes. And out of the Krebs cycle, you make what? A lot of NADH, right? I don't even need you to know how many, and a lot of FADH too. Right? NADH and FADH2 gets made here. There's some ATP, right? 2 ATPs, but do we really care about these 2 ATPs? Yes and no. Yes, they're being made, so that's good, but really the important part is what? The electron transport chain. And these two here, these are doing what? They're transporting electrons to the electron transport chain, where you get, again, chemiosmosis. You get that movement of electrons across the barrier, the membrane, to create a high and low hydrogen ions. Why do we do that? So that we can make, finally, over here, we can do what? We can make ATP. ADP goes to ATP. And of course, we have to remember always, as hydrogen ions go through, always we have to remember, it's a really big deal that you remember that oxygen gas is the last electron acceptor, and you make water. If you know that, all that, you'll be fine. How am I going to test it? Remember the practice test you took and all the other questions. If I tell you that I fed a gerbil a sugar molecule whose carbon had, hear my question, whose carbon was radioactive, what molecule in this whole, everything you see on the screen, what molecule is going to be radioactive after you've done respiration? Listen, I feed radioactive sugar to a hamster. I let it do respiration, and I measure I'm trying to find where that carbon is with a Geiger counter. Where am I going to find it? What molecule?
Oxygen.
Not oxygen.
Oh.
Not oxygen. See, you saying oxygen means you're not thinking. The oxygen you breathe in doesn't come from sugar. You're breathing it in. I'm telling you, a gerbil's not doing photosynthesis. It's going to be in the carbon dioxide. That's right. Now, you guys are thinking about the problem I asked you on Monday, which is, what if I made radioactive CO2 and I fed it to the plant, right? I let the plant take in radioactive CO2. The oxygen in CO2 was radioactive. Where would I find that radiation, and what molecule in photosynthesis would I find it? Not oxygen. Because the oxygen from photosynthesis comes from what? From the water. I would find it in sugar or any of the steps in the Calvin cycle, because that's where CO2 comes in, right? All right, so that's the kind of thinking you have to get in your head. You have to think, okay, well, if he's asking me about carbon dioxide, if he's asking me about sugar, if he's asking me about oxygen, where did that molecule come from? Did it come from the light reactions, the Calvin cycle? I know I'm making sugar in the Calvin cycle. I know I'm making oxygen gas. If I give you radioactive water, heavy water, then to a plant, where would I expect that radioactive oxygen to be? In the gas, the oxygen gas. You see, if I make this radioactive, what does it get turned into? Oxygen gas. So that's the kind of thinking you have to use. All right, have a good one.